Hey everyone, in this video I am going to describe the virus known as HIV-1 because scientists and physicians have successfully used certain aspects of HIV-1 in gene therapy. And we're going to learn a little bit about HIV-1 and hopefully that's going to help us understand how this certain gene therapy approach called or that uses lentiviral vectors works. So by learning about HIV-1 will be, it'll be easier for us to understand how these lentiviral vectors work in gene therapy. And what's the connection here? Well, HIV-1 is one of the, I think, only two known lentiviruses, types of viruses called lentiviruses. So one of only two types of lentiviruses in humans. The other one is HIV-2, which is less common than HIV-1. So HIV-1 is really complicated. And I will try to make it not that complicated for us to understand. So the HIV-1 genome is 9.7 kilobases, so 9,700 base pairs. I'm going to talk about the Provirus first. And Provirus is the word we use to describe the HIV-1 genome when it is integrated into, say, the human genome, so in the DNA state. When it's part of the virus, the infectious viral particle, it's an RNA genome. So we like to say HIV-1 has a single-stranded RNA genome. But first I wanted to describe the provirus state, which is a copy of the HIV-1 genome in, let's say, in the DNA state, inside, say, the human genome. So in the provirus state, it will be flanked by target site duplications. Now, after integration, the target site is duplicated. That is 5 base pairs. And on the ends, there will be these regions called LTRs for long terminal repeats. Now, unlike what we have already seen for our class 2 DNA transposons, where the TIRs, so the terminal inverted repeats, are identical sequences that are inverted relative to one another. These LTRs are direct repeats. So they're not inverted relative to one another, they're just duplicated at the ends, and they go in the same direction. And I think it's something about 336 base pairs long. So the HIV-1 LTRs are 630 base pairs long. Now let's just say about 650, just in case that number's not correct. So about 650 base pairs long. Now, let's say everything between the LTRs. Okay, so there's a lot of stuff in here. And researchers have defined 9 genes. Now, genes, this is where this word gene starts to, break down our definition of what a gene is and isn't. It starts to break down here because it's really confusing. And I'm going to talk more about that in a moment. But researchers have defined 9 genes. And we're going to call, I'm going to name them for us. So GAG, POL, ENV for envelope, REV, R-E-V, TAT, NEF, VIF, VPR, and VPU. So let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9. So there are our 9 different genes. And the GAG gene here encodes 4 different proteins. So there are going to be 4 proteins. I guess one long polyprotein will be made, and that polyprotein will be cleaved into 4 different proteins with important functions. Similarly, the POL gene encodes 3 proteins, ENV 2 proteins, and the rest of these are all just single proteins from the rest of these right here. So what does that give us? So 4, 3, 2. So 4 plus 3 plus 2 is 9, 10, 11, 12, 13, 14, 15. So at least 15 important proteins are encoded by the HIV-1 genome. Now, depending on what reference you use or what researcher you talk to, there may be more than 15 proteins. There are these little peptides that are released when, say, these longer polyproteins are processed into smaller proteins, and I think some researchers consider those to be important proteins with functions, so the number could go up. So I'm going to settle on at least 15 proteins. Now, when we start to talk about the parts of HIV-1 that are needed for gene therapy, that are needed in this to make a lentiviral vector, we're only going to need these up here. All of these down here are dispensable. So it's taken, I think, decades, but researchers have figured out a way to make viral vectors using just these up top here. So we're going to focus most of our discussion on the proteins that come from these 4 genes right here. Now, I wanted to talk a little bit about these diagrams here, because if you're new to HIV-1 biology, it can be really, these diagrams can be really confusing and difficult to interpret. So let me start with just a similar diagram to what I have already shown in on the previous piece of paper. And I want to put the LTRs here. Now I'm going to show the locations of these genes. So in the notes, you'll see one of these diagrams, and there are many variations on this. Essentially, what we're doing with a diagram like this is showing the locations of the coding regions for the genes I just went over. So about this part of the HIV-1 genome, this is where we're going to find the coding region for the GAG polyprotein. So over here would be the coding region for the POL polyprotein. Now we can see these overlap. So that happens, a lot of the genes here are going to have overlapping regions, which makes it hard to define, like, okay, where does one gene end, what does one gene start, what makes a unique gene. So very difficult to define a gene when it comes to the HIV-1 genome and all these different important sequences. So we're not going to worry too much about the definition of a gene. We're just going to go with what, the researchers generally agree that, okay, there are 9 different genes in the HIV-1 genome. And we already went over the names of those. So we have some overlap here. We have some overlap here with VPR. And then there are some introns that are removed to give us messenger RNAs that can give us the REV protein and the TAT protein. This is an approximate diagram, right? The exact positions of these overlaps might be off. And ENV is going to be right about here. VPU overlaps here with ENV, and NEF is going to be here. So let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, we have the approximate locations of the 9 genes in the HIV-1 genome. So now these are the 9 genes, and there aren't 9 transcripts though. So there are, I think, at least 7 transcripts, and we're only going to discuss 3 of them. So it might help if I just diagram some of these transcripts. And so what do I mean by transcripts? Well, if this is HIV-1, start with the original diagram. the LTRs. Now, this needs to be transcribed to make a messenger RNA that could be translated to make those proteins, right? So there's a transcription start site in this LTR. We're going to call this the 5' LTR. There's a transcription start site, let's say about 1/3 of the way into this LTR. And then there's a stop site. Well, the poly A addition site is going to be in this 3' LTR. So RNA polymerase II can bind in here inside the LTR, start transcribing, and it's going to give us a transcript with a 5' cap. Again, we haven't talked that much about 5' capping of transcripts yet. We're going to in the next section of the course. But it can transcribe this whole thing and give us a poly A tail at the poly A site. Now this RNA transcript, starting here, ending here, with no removal of any intronic sequences, this is called two things. This is the genomic RNA. So this is the RNA that's going to be incorporated into the virus particle, but it also serves as a messenger RNA for a few of the different proteins. So let's say there's a start codon here, and we have a ribosome that binds in the 5' UTR of this messenger RNA, and there's a stop codon here. So translation of this fragment, this segment, interval I should say, of this messenger RNA will give us that GAG polyprotein. This part of the protein is going to be what we're going to call MA for the matrix protein. This part right here is going to be CA for capsid protein. This part is going to be NC for nucleocapsid protein. And this one is going to be P6. It's important for budding of the virus particle from the cell membrane. Now, these are all linked together, and they will need to be cleaved, separated from one another by a viral protease, which I'm going to talk about in a moment. So something very interesting happens. So this happens about 95% of the time that this type of messenger RNA, this full-length messenger RNA, is translated by ribosomes. 95% of the time we get this. 5% of the time we're going to get a ribosomal frame shift, -1. So what that means is the ribosome is going to back up just one nucleotide. So -1 ribosomal frame shift. That's going to cause the ribosome to, that's going to eliminate this stop codon, because the stop codon is no longer in frame if the ribosome backs up one position and starts translating. And the ribosome is going to move and translate everything down to about this position down here. So because of this frame shift, which happens just somewhere right before or close to this stop codon, the ribosome backs up, it changes the reading frame and moves forward. So there's no stop codon here, and we end up with a slightly different protein starting at about this position. Slightly different polyprotein, I should say. So MA, CA, all of those are still okay, but this one right here I'm not that sure about. I think P6 is lost, completely lost because of the frame shift, and then what we get is PR for protease, RT for reverse transcriptase, and IN for integrase. Now these right here, these are the proteins made by that pol gene. These right here, these 4 and these 3, these are the GAG associated proteins. Let me go back here. So we see how we have these GAGs and pols right here overlapping. Well, the whole thing right here, this whole thing right here, this polyprotein is called GAG pol. And this polyprotein right here is going to be called, what is it called again? It's called PR55 GAG. I'm guessing the polyprotein is 55 kilodaltons or something. That's my guess. So, okay, now this is the protease right here that's going to help separate all these different proteins here and up here. Now, okay, so I wanted to mention this because these are really important proteins when it comes for understanding how the viral vector works. I just want to talk about two more transcripts. So as far as I know, all of the transcripts start at this same site right here, but we have some alternate splicing that can happen. So this is the one full-length transcript. So there's an intron that can be removed, say between here and let's say about here. And a messenger RNA, an HIV-1 messenger RNA with this approximate splicing pattern can be used. Let's say the ribosome starts here. Let's say there's a start site here and a stop site here. Can be used to translate the ENV gene, which gives us proteins called GP120, sorry, and GP41. Now these are going to be the envelope proteins. These are going to be important for helping the viral vectors fuse with a host cell. I'm going to talk more about these in a moment. So this is GP120, this is GP41. And just one more. So intron, this first, the long RNA is transcribed. This little piece is removed as an intron, and then another one down here is removed. And again, ribosomes kind of entering at the same place right here. And there's a start codon here, and the stop is going to be down here. And then this translation of this messenger RNA here, removing these two introns, is going to give us the Rev protein. And the Rev protein is another one that we need for the viral vectors to work. Okay, so in the notes, I'll give you some more details on all the different transcripts. The important thing to understand is that there's some alternate splicing that can happen. And depending on the way the transcripts are spliced, the exons are spliced together, we can get the different HIV proteins that are needed for a productive infection. And my battery is about to die on my phone, so I just want to go over a few more things, and then you can watch the video on the reverse transcription of HIV-1, and the final video for this lecture will be on how HIV-1 integrates into the genome. So this is my diagram that I pieced together from a bunch of different sources. So HIV-1 virus. Okay, so we have the membrane, or the envelope. We're going to call this the envelope. So the viral envelope. And the viral envelope is derived from the infected cell through a process called budding. So when the virus buds from the cell, it takes some of the host cell membrane with it. These are the envelope proteins, GP120, GP41. So these allow the virus to infect a specific type of cell. So I'm going to talk about that in a moment, but they interact with a receptor on certain T cells. So they allow the virus to infect T cells, and that's why HIV-1 infects T cells, and it leads to leads to AIDS, acquired immune deficiency, if I remember correctly. So this right here, this purple, this is going to be that matrix protein, MA. This right here, this is the capsid protein. Now, I'm only showing one RNA molecule in here, but HIV virions, virion being the infectious particle, has 2 copies of the HIV genomic RNA in there. I only diagrammed one. And this red thing right here, well, that's going to be what we call the nucleocapsid. So we have capsid, which is the blue protein that sort of, it provides a protective shell around the RNA genome. The nucleocapsid is the protein that sort of binds directly to the RNA and protects it. Now this yellow, that's going to be the integrase. That blue right there, we're going to call that the reverse transcriptase. And these oranges right here, these orange things, these are going to be the proteases. So the virus has a bunch of the proteins in it that are needed to infect the cell and integrate or reverse transcribe its RNA and integrate it into the host genome after turning it into DNA, right? So just a little bit on how the infection process works. Okay, so we have the HIV-1 virion, the infectious particle, the envelope proteins, GP120 and GP41, interact with receptors on T cells, the CD4 receptor, and I believe this one is called CRC5. Now, when they interact, this causes the virus to insert the cell, just like I've diagrammed down here. So these membranes fuse, and you can see this the way that the membranes are fusing. This capsid with the viral RNA inside it is released into the cytoplasm. Now, in recent years, researchers have come up with a model that proposes that this whole thing right here is transported through the cytoplasm, possibly with the help of filaments or microtubules. So transported throughout the cytoplasm and through the nuclear pore. Well, this is the nuclear pore. This is going to be the nucleus down here. This is the cytoplasm up here. And when this gets to the nucleus, then the capsid is dissolved, and then we'll have the RNA here, which can undergo reverse transcription, and then that can be integrated into the genome. Now, this is why researchers like lentiviruses for inserting DNA into genomes, because it has the ability to go through the nuclear pore. They can introduce DNA into the genomes of non-dividing cells. So some viruses that can insert DNA into dividing cells only, they can do that because, even though they can't go through a nuclear pore, they can wait until the cell undergoes mitosis and the nuclear membrane dissolves, and then they can get at those chromosomes. But lentiviruses, they have the ability to go through the nuclear pore, so they can get DNA into the genomes of both dividing cells and non-dividing cells. Non-dividing cells have the nuclear envelope, the nuclear membrane, so the viruses need a way to get through. And lentiviruses like HIV-1, they can go through the nuclear pore. So now I'm going to talk about how the RNA is reverse transcribed in the next video, and then I'm going to talk about how it is integrated into the genome in the video after that. For now, I want to jump to, okay, assuming it's reverse transcribed, assuming it's integrated into the genome. Okay, so we went over how the genome is transcribed into different messenger RNAs, how the genomic RNA is made. I just mentioned that 2 different genomic RNAs, well, identical genomic RNAs, are incorporated into the virion. So here, what I'm showing you is how the budding process works, how the proteins are transported to the host cell membrane. So this is an infected cell, host cell membrane. And the virus has integrated into the genome, and it's being transcribed, and it's making new viral particles. So new viral particles are being, assembled and are budding from the cell so they can go infect other cells. So we talked about those polyproteins before, right? So let me show you. Right, so this is the GAG polyprotein. This is the GAG-POL polyprotein. 95% are going to be like this. 5% are like this. So these go to the membrane, and they look something like this. You can notice that they haven't been cut into individual units yet by the protease. So here we have the long one, the GAG-POL protein here, and we have another one over here. And these interact with the RNA, so they help transport the genomic RNAs to the cell membrane, so the genomic RNAs can be incorporated into the virus. So in the envelope proteins, they also are transported to the membrane. They insert in the membrane. So you can see as this starts budding from the host cell membrane, we're going to have all these proteins inside the virion. It's going to be the immature virion at first. Now after budding, there are some changes that happen. That protease starts to remove or break all the separate all these different proteins to make the mature virion. So that's when we go from the immature state, the protease hasn't done its job yet, hasn't separated all the polyproteins, to the infectious state where we have something like this. Okay, now this virus can go and it can infect another cell. So I hope you understood. That was just a brief, somewhat, well, I thought it's probably not too brief. I probably could have made it briefer, but I think I covered all the essential things we need to know about HIV-1 biology, except for reverse transcription, which I will cover in the next video, and integration, which I will cover in a video after that. But once we cover all that, then it should be really easy for us to see how HIV-1 based lentiviral vectors can be used in gene therapy. Okay, see you in the next video.